Welcome to Beekeeping Today podcast presented by Bee Culture. Beekeeping Today podcast is your source for beekeeping news, information, and entertainment. I'm Jeff Ott.
And I'm Kim Flatham.
Hey, Jeff and Kim. Today's sponsor is Global Patties. They're a family-operated business that manufactures protein supplement patties for honeybees. It's a good time to think about honeybee nutrition. Feeding your hives protein supplement patties will ensure that they produce strong and healthy colonies by increasing brood production and overall honey flow. Now is a great time to consider what type of patty is right for your area and your honeybees. Global offers a variety of standard patties as well as custom patties to meet your needs. No matter where you are, Global is ready to serve you out of their manufacturing plants in Airdrie, Alberta, and in Butte, Montana, or from distribution depots across the continent. Visit them today at www.globalpatties.com.
Hey, thanks a lot, Sherry. We also want to thank Bee Culture Magazine for continuing their presenting sponsorship of this podcast. Bee Culture has been the magazine for American beekeeping since 1873. Subscribe to Bee Culture today, and while you're there, check out Bee Culture's Beekeeping, your first 3 years, a quarterly magazine for beginning beekeepers. We also want to thank 2 Million Blossoms as sponsor of this episode. 2 Million Blossoms is a quarterly magazine dedicated to protecting all pollinator insects, both wild and managed, before they disappear. Learn more in our Season 2, Episode 9 podcast with editor and our guest co-host Kirsten Trainer, and from visiting www.2millionblossoms.com, and that is with a number 2. Hey everybody, thanks for joining us again. Kim, we have a special guest lined up for today's episode, Dr. Samuel Ramsey.
Yeah, Jeff, we're lucky we caught Sammy this time. He was over in Thailand studying that another nasty mite over there. Yeah. And his research got interrupted with the pandemic, so he had to come home. So we caught him when he was here.
Yeah, I guess it's a way to make, what do they say, lemonade out of lemons. As we'll hear that, his shutting down his experiments and everything coming back because of the pandemic has worked out in our favor, getting him back on the show. He was on our show back in 2018. It was a great interview, a great show. Sammy is quite entertaining and a friend of the podcast. So we look forward to having him here.
See what he's found out about Tropilaelaps so far.
No, if you look at him on the internet and YouTube him, well, you'll see why he's so focused on stopping them before they get here. So how's your summer going, Kim? You're usually doing a lot of meetings and
I'll tell you, Jeff, this is the summer of Zoom and meetings, virtual meetings. I know some people are still having meetings in person, and they're trying to do all of the right things, but by far the most have gone to, and most of them are using Zoom, as a matter of fact. I've given 3 talks in the last couple of weeks, as many as 150 people and as few as 40. But it's always Zoom. And if your group isn't using this program or one similar to it, you might want to think about joining the present. And beekeepers tend to be a little slow in picking some of these things up. I was, and Jeff got me going, but if you belong to an association and you're not having meetings, consider figure out how to do Zoom.
Yeah, it's obviously it's saved the world. The world runs on Zoom right now, and they've gone from when we started using a small obscure little video conferencing. Now it's a verb, an adverb. Yep. It's a noun. It is. People are Zooming, so yeah. Good words of advice. All right, well, let's not hold this off anymore. Our interview was Dr. Samuel Ramsey. Very entertaining. We hope you enjoy it. Well, Dr. Samuel Ramsey, welcome back to the Beekeeping Today podcast. I'm happy to have you here.
Oh, it's great to be back.
Sammy, it's good to see you again.
Good to see you too, Kim.
I think we crossed paths at Apimundia, didn't we? I think across the room I waved.
Oh my goodness. I'm going to have to figure out something new for these conferences and things. I feel like I am just zooming around to every different booth, to all of these different talks, and I don't get the opportunity to really invest in conversation the way that I would like to. It's been ages since I seen you, and it still feels like it's been ages since I've seen you, since I've just seen you at Apimundia, because we hardly got to talk.
Well, certainly understand that meeting. That was hectic in capital letters, bold underline.
Yes,
but,
sir.
It was fun. It was a good meeting. So, Sammy, you have been in Thailand.
I have.
Since we talked to you last.
And a few other countries.
And you have been busy studying something we all need to worry about. Why don't you share with us what you've been up to and what we need to know?
I certainly will. So I've been studying a parasite that I actually mentioned the last time that I was on this podcast, and it's the Tropilaelaps mite, Tropilaelaps mercedes. And given that the name that I just said is one that few people can repeat back to me after hearing it once, kind of there's somewhere out there, like I somewhere deep down inside, I've got my fingers crossed that whoever came up with the name Murder Hornets might find a good moniker for Tropilaelaps mercedes, because it needs better marketing. I don't think people are.
The Tropilaelaps is fine. The mercedes is the tongue twister.
Well, I don't feel like they've quite got the marketing that they need to have. So this is a parasite that, a lot like Varroa, it's in the ways that it interacts with a honeybee colony. There is an intimate association there. There's a connection with the development of the honeybees. When they are at their most vulnerable, those mites are going in and causing all kinds of damage. They're pretty much Varroa destructor with some steroids thrown in, just kind of the bodybuilding cousin. And I'm a little bit concerned that there's so little that we understand about this parasite currently, because were it to arrive in the U.S., there's still so much work that would need to be done for us to really grasp elements of its impact, its biology, its life cycle, and how best we can treat these organisms.
Can you describe the Tropilaelaps? Just compare and contrast, because I think a lot of people are familiar at this point with Varroa.
So Tropilaelaps mercedes, like Varroa destructor, comes in a few different varieties. So Varroa has 4 species, and right now we know 4 species of Tropilaelaps. It is a mite, a lot like Varroa, but about a third the size, so more difficult to see. And it's sort of shaped like a pill with some very long legs all around it. And it's a very, very fast parasite by comparison to Varroa. So Varroa is this reddish, weird oblong-shaped parasite that just sort of bumbles along and moves slowly. Tropilaelaps is a pill-shaped organism that speeds along on the comb, running around back and forth. It's rather conspicuous to see in colonies and causes some rather conspicuous damage. Just months into an infestation of Tropilaelaps, you'll start to see tons and tons of the cells being uncapped as the bees go through and attempt to control a problem that looks a lot like parasitic mite syndrome in some ways. So they're going through, they're uncapping the cells, but unfortunately, with a lot of this uncapping that's done, they don't actually kill the parasites inside. In my time in Thailand, I've found that the parasites seem to survive this process just fine, and oftentimes continue reproducing just as they would have otherwise, as the bees are dragging more of their developing brood out. Tropilaelaps is also interesting because of the small amount of time that it spends on the adult bees. So Varroa can spend anywhere from 1 day to a couple of weeks on adult bees, with about an average of 3 days. Tropilaelaps spends usually less than a day on adult bees. So it's really more a matter measured by hours before it goes right back into its reproductive cycle. So even though it doesn't reproduce much faster than Varroa in terms of its ability to produce eggs and go through the reproductive process, its population builds up so much more quickly because they don't have that lengthy refractory period that you see with Varroa. In addition, that makes them a lot more difficult to control, because the time that these parasites are on the adult bees is the time where they are the most vulnerable, and Tropilaelaps has been smart enough to reduce that amount of time to as short a period as it possibly can.
Wow.
Sammy, this is what you just alluded to, kind of, is that the life cycle is similar to Varroa.
Yes.
A gravid female goes into a cell, waits until it's just before it's capped, produces male and female offspring. The male and female offspring mate, and then if things go the way they should go, the larva emerges and they emerge with it?
Precisely. And thank you for that, Kim, because I forget sometimes that it was a good save there. I forget sometimes there that everybody doesn't just immediately know the life cycle of a Tropilaelaps mite. When you've been immersed in these things for long enough, some of these things, they just stand out to you like, oh yeah, no. Everybody knows they jump in there right before the cell is capped. They reproduce at a regular rate, and the cell is uncapped by the adult bees, and the mites go out with them. Of course, yes,
everybody knows
everyone
that.
knows that, right? No, it's precisely as you said. So Varroa's life cycle, you've just kind of given the rundown for what is the typical Varroa life cycle, where the parasite will sort of ride around on adult bees on the underside of their body for the most part when they're trying to find a new host. But the entire reproductive process happens under that wax cell capping. And when they're under the cell capping, they feed very regularly on the developing brood, which is awful because the brood is totally incapable of defending itself. And so it's just sitting there as this organism feeds on it, has offspring, and then the offspring feed on it as well through that same hole. So you go from having one mite feeding there to an entire family as they develop. And then as the bee finally turns into this adult that's had a lot of the nutrition removed from its body when it needed it most, and it chews its way out, the mites are then able to leave. And if there are any adult daughters in that cohort, they're able to carry on this process again. They leave already mated. And so it makes it a lot easier for them to continue their reproductive processes within that colony or to be flown to another one without having to mate again.
That's Varroa and the Tropilaelaps. Or are you just...
So that's Varroa. Yeah. And I've described the elements of it that are similar between the two of them. But there are some key differences because when Varroa goes into one of those cells, it lays an egg, and that first egg is always female. And then the next egg, the first egg is always male, and the next eggs are all female. And so that one male, his only job is to reach adulthood before his sisters and make sure that he's able to mate with them so that they can emerge already mated. This is probably the part where we should warn anyone with small children. This is not quite the section of the podcast they want their kids listening to.
We'll have to put an explicit label on this podcast.
Yeah. So after that bout of parasitic incest, those females can then emerge from the cells and they can mate with, they can carry on the processes reproductively without ever having to mate again. With Tropilaelaps, the interesting thing about it is that when the female enters the cell, her egg can be a male or a female. And she can alternate between male and female eggs throughout the process. And one of the reasons why she doesn't have to make sure that first egg is a male is because she's not racing the clock. She doesn't have to make sure that those females are mated before the cell is uncapped because the males are actually capable of leaving that cell as well. For Varroa, when the cell is uncapped, the males are not fit for life. Their bodies don't have that hard coating over them that Varroa females have. And as a result, the water evaporates from their body really quickly and they just die. But the male Tropilaelaps are able to leave the cells and they avoid something really problematic that you just heard from me a moment ago. There's a very strong limit to the amount of genetic diversity that you're going to see in Varroa because they're always mating with their brothers. Now, there are circumstances where two female Varroa will enter the same cell and produce offspring, and then the males produced there have the opportunity to mate with females that aren't their direct sisters. But most of the time, they are only mating with their siblings. For Tropilaelaps, those males are able to leave and mate with any females that they want to, creating much more genetic diversity and allowing them to have a robust genome that's allowed them to develop pesticide resistance to chemicals that have only been used on them very, very briefly. And so if you look at the genome for Tropilaelaps, if you actually attempt to test pesticides on them, you'll find that they're already showing resistance to some of the best chemicals that we have at our disposal currently.
Wow.
I have to ask this because of our guests, Jeff. When these mites are feeding on these larvae, honeybee larvae, if I recall, the Varroa female starts on a spot, and then all of the offspring feed at that same spot.
That's
and
accurate.
is that where that spot is on that larva, is that where the fat body on that larva is?
So that's the larva. That's a great question, by the way. The larva is pretty much all fat body. It's kind of ridiculous, but it is a sack of tons of fat body, a small brain, and a digestive system running through the middle. And they need so much fat body because the fat body is doing so many important processes that are necessary for metamorphosis to happen. Because if you think about it, metamorphosis is doing some really, it is a complicated process to get you from being just a sac with a mouth on one end and a rectum on the other end to being an organism with wings, functional eyes, organs capable of mating, this much larger, much more convoluted brain. All of that reorientation is happening because the fat body is able to facilitate this process. And so you need a lot of fat body tissue. It's also there storing nutrients. So when the mites bite into a section of the bee, what's most important for them is that they don't bite the head of the bee, because if they cause a deformation in the head, they can kill their host. And if they kill their host, it can't chew its way out of the cell and allow them to be released because they don't have the apparatus to do that on their own. And so what you see from them is that as their preference is for them to then feed, usually on the underside of the mite's body. They'll feed in a section there that's destined to be the abdomen. But if they chose to feed on other regions of the mite's body, they would still be able to contact the fat body tissue. But something else that you've brought up there that I think is really important to say is that they have a fairly polite manner about them as parasites. You know, they've decided we're going to feed on this one spot, and our entire family is going to feed on this one spot, and we're going to try not to disturb other elements of the organism's development and body. Tropilaelaps have no element of those manners. They will feed on every little spot on the developing bee's body, and every time they feed, they bite a separate hole. So while Varroa only feed through the same hole, Tropilaelaps keep feeding through different sections of the bee's body, causing all kinds of damage and deformities in the process.
Well, that sounds like then if the bee is even able to emerge, it is far less capable of becoming a bee, of contributing to the health of the hive and living.
Yeah, you want it to be a functional member of society. You want it to be capable of eventually being a forager and bringing back food, a nurse, to then create food for the brood. And unfortunately, when they have been fed on by Tropilaelaps, the regions of their body where they're fed on, there's a section that can kind of develop like scar tissue. And so you'll have this area of the bee's body that becomes inflexible and rather rigid. And so if that happens near a joint, the bee won't be able to bend its leg, and it won't be able to put its leg down and actually put pressure on it. If it happens near where the wings are, you'll get these odd deformities in the wings. And this is to the exclusion of viruses. This is without viruses present. They can cause deformities in the bees themselves, and that's concerning. So Tropilaelaps is not something that I ever want to see in the United States. And it is my sincere hope that it never shows up here. But we also have to be vigilant as researchers, as beekeepers, to make sure that we are keeping an eye out for this organism should it ever arrive, and to make sure that we have a plan for what we would do when it arrives.
I'm going to go back to what you were just talking about. You mentioned virus, and is Tropilaelaps spreading virus like Varroa do?
Tropilaelaps is capable of spreading viruses like Varroa, which is unfortunate. We have found deformed wing virus that is a very competent vector of deformed wing virus. It is also a competent vector of black queen cell virus. So far, those are the only two that we know that they are capable of vectoring directly. However, there are several other viruses out there that they have not even been tested for. And so we really aren't sure. There are a lot of questions with Tropilaelaps that have not been answered.
Do they have
So they're bad and they could be worse.
Do they have a preference in larvae that they feed on? Like the Varroas that go to the drone?
Chef.
What?
That is a great question.
I thought I was in trouble.
So
go ahead.
Well, I had to throw that out there because there was something that there are a few things that I wanted to say about this organism, and the two of you have just been helping me move right along and saying it, so I appreciate that. So the differences in the life cycle, but also the differences in their preferences. I've read a few papers, and I've even been part of a review where we have discussed elements of this mite's life cycle, and unfortunately, as we have discussed these themes, we've been heavily reliant on papers that have based their understanding of this parasite on Varroa's life cycle. A lot of what's been said is this parasite is a lot like Varroa, and so we've assumed a lot like Varroa, it does this or it does that. It must be more interested in drones than it is in workers, because workers have a shorter developmental time, drones have a longer developmental time. It would simply make sense if Tropilaelaps were more interested in drones. And so I've seen that in papers, and unfortunately, I've been one to even repeat that I've read in papers they're interested in drones. But in my time in Thailand, actually researching this organism, I've found I've seen no support for that in my own work, and I've been steadily collecting more and more data on this subject, and it seems like they do not have a preference for drone larvae, and that may be one of the reasons why they are so damaging so quickly to the colony, because and this also might be difficult for people to hear, but the men in the colony aren't really doing much. Like they have a purpose. They are a part of the divine plan for bees, but they don't do very much inside of the colony. And so if you are going to cause substantial damage to the colony, you damage the workers. And when there aren't that many drones in the colony and Varroa move into the worker brood, and the worker brood is all that they have access to, they can cause pretty substantial damage pretty quickly without that buffer of drones to soak up some of that damage. With Tropilaelaps mercedes, we find them in the drone brood, we find them in the worker brood, but I've found colonies that have had greater than 40 or 50% of their worker brood infested with Tropilaelaps mites. And you cannot have a functional colony with a parasite that damaging, constantly destroying your developing offspring.
Wow.
Well, it begs a question, Sammy. How are these people keeping bees?
It begs an uncomfortable question about how these people keeping bees. So I will say I've been working in Thailand for several months to try to better understand this parasite. And this was a study funded to specifically answer the question that Kim just threw out there. Beekeepers have funded this project. And through the help of beekeepers, we raised enough money for me to be able to go to Thailand and understand this organism. And something that I've wanted to understand is, how are bees existing with Tropilaelaps present in those colonies? How are beekeepers making a living, producing honey? How are they doing anything with these parasites present? So I will tell you now that the news isn't wonderful here, but beekeepers do a lot of walk-away splits in areas where Tropilaelaps is present. A lot of what I've seen in Southeast Asia is a constant splitting of the colony and a removal of the brood, which is going to very much slow down the progress of the colony, the buildup of the colony, and the productivity, like how much honey they're capable of producing, how much wax. If you're interested in their wax production, all of that takes a hit when you're constantly removing the next generation from this process. But when they see the mites begin to build up, it becomes imperative that they get rid of what the mites need in order to complete their life cycle. And so they remove the brood, and you have these broodless colonies that have to build back up again. And that gives you a few months of buffer time to play with, and sometimes just a couple of months of buffer time to play with before you have to do it again.
Wow.
That makes perfect sense. When what you said earlier is that the time that they spend outside of the cell is very, very short.
Exactly.
So if you go queenless for 21 days or whatever, you've got a really an advantage there doing that. Of course, you don't have any more bees being produced, but at the same time, you have how much of the a mildly infested colony and you do a walk-away split and you let them raise their own queen, and that takes, by the time she start laying, 24, 25 days?
Mm-hmm.
How many of the mites will perish in that time?
In that time, you can expect that virtually all of the mites in that colony are going to perish, because they do not seem to be able to survive for more than a couple of days without brood. On the adult bee population, it's very possible that they're simply not able to feed. Varroa destructor has a body that is designed specifically for them to be able to wedge themselves between the plates that make up the bee's body so that they can access the most nutritively dense tissue in the bee's body. They get a lot of nutrition by being able to climb onto the abdomen, go between the plates of the abdomen, stick their mouth parts there, and feed on that bee's fat body. Tropilaelaps don't have that ability. They're shaped, they have this odd pill shape to their body that doesn't allow them to wedge between the plates. It also doesn't allow them to hang on very well when the bees are in flight. And so they don't hang on well to foragers. They don't seem to do well hanging on to nurses, and they don't seem to be able to feed on them. So they're pretty useless in this phase when they're on the adult bee population. And that's part of the reason why it seems like they are moving in the direction evolutionarily of eliminating it altogether. But it's not something that just happens in one quick bout.
Well, there is some refreshing news. There is a natural defense that beekeepers can use without having to use poison.
That is very true.
Do they use any poison over there?
They do. So there are several different chemicals that are used, but unfortunately, a lot of them are too expensive for most of the population to be able to afford on a regular basis. And so people have been experimenting with a lot of oils there, which fortunately are less damaging to the bees in a number of ways. But lemongrass oil is one chemical that is used there. The one that I've been both fascinated by and perplexed by was the usage of formic acid there, because a lot of the beekeepers have told me that their method for controlling this parasite is to actually take these wooden paint stirrers, so the same kind that you can get from like a Home Depot or a Lowe's. They'll take these wooden paint stirrers, soak them in industrial grade formic acid, which you can get from these shops that supply plantations. The rubber plantations there use it to cause the rubber to congeal. They soak these sticks in this very concentrated formic acid. It's so concentrated you cannot be near this stuff. When you open the cap, it will burn your eyebrows off. It's very concentrated stuff. They soak it overnight and then slip it into the front of the colony. And I have a video that I showed of myself and a couple of people who I was working with who were inserting these into the fronts of colonies, and the immediate reaction that you get from the bees. The bees hate it. They fall all over themselves to get out of the entrance of that colony. The second you slide those things in there, it looks ridiculous, but it is effective because you're not so worried about the adult population of bees. The adult population of bees is getting out, but the brood that's stuck in there, they are constantly being exposed to this vapor that is functioning as a fumigant and is able to penetrate those wax cell cappings, get in there and actually kill the mites below the capping. And I've worked very hard to actually verify this. And so I was going through in my 36 colonies in Thailand every single week. We were going through and taking a frame out of the colony, looking at the number of mites that were dead below the capping. And I've got some pretty solid data. We weren't able to finish the full data set because unfortunately COVID-19 had other plans for where I would be this year. But we're able to see that this formic acid works very effectively to kill the mites below the capping. Unfortunately, it is just harsh on the bees themselves. Very harsh. You can kill, because the dosage itself is not regulated in this. If you have wood that happens to be a bit more porous, it can absorb more of the formic acid because the release of it is not in any way standardized. Different levels of heat can cause it to be released more quickly or more slowly. You just really don't know what you're getting in just sliding these paint stirrers into the colony, but it does work.
How long do they leave them on?
just takes about a day, and usually they're spent after that. Mm-hmm. Not that I am recommending this to anybody, by the way. As a United States Department of Agriculture researcher, I had to make it very abundantly clear that I am not recommending this to anyone. We do actually have options at our disposal for the application of formic acid that are standardized, that are substantially less dangerous to us and the bees. And it would be much wiser to have those at your disposal. They're just more expensive overseas and actually aren't licensed.
And we're talking about Tropilaelaps too. So it's, yes. Fortunately, we're not dealing with that yet.
I was going to mention that, though. We do have a routine that works with our bees and our mites that we could probably easily, well, I don't know about easily, but it would certainly work for both at the same time.
Well, now, unfortunately, I'm gonna have to problematize that particular assumption there, Kim, because I like that you're thinking about that. What could we do quickly if Tropilaelaps arrived, where we'd be able to potentially eradicate it or control it, at least bring it under control? The problem is that even though the differences between its life cycle and Varroa's life cycle are subtle, they make all the difference. The fact that, so we wouldn't be able to use the typical drone trapping measures that I've discussed earlier because they don't have that attraction to the drones. But what about other methods that we could potentially modify? Maybe we could use amitraz. Turns out, it seems like they've actually developed some level of resistance already to amitraz, and it's not an effective measure to begin with because amitraz targets the population of the mites that are on the adult bees. And so I've been going through and looking at more and more and more of our measures that we would potentially attempt to modify from Varroa to use against Tropilaelaps. And it turns out the vast majority of what we use in terms of chemicals target the population on the adult bees. We have very few measures at our disposal that are capable of penetrating the capping and going after those mites. And that's the reason why I was testing formic acid while in Thailand and looking at its effectiveness, because that would likely be one of the best options at our disposal, or at least it's something in our toolbox that would potentially have some effectiveness against this parasite. But we don't have a lot of other options aside from that. Now, something else that I also tested in this process was these heaters that can actually be used on the colonies. They can heat the colonies, and as a result of this difference between the amount of heat that the adult bees are capable of dealing with and the amount of heat that these mites are capable of dealing with, there's a difference there of several degrees. And so that gives you something to play with where you can actually kill the mites without killing the brood or the adult bees. And that is also an effective means of impacting these mites. I am still collecting data on this and will need to return to Thailand in order to finish this project. But preliminary work shows that both of those options show some level in killing these parasites. And I'll have actual numbers for you the next time you see me.
That's amazing.
You've been doing your work in Thailand. Where else are these mites?
Oof. So the mites are distributed throughout Southeast Asia, and that was their original location in the world. Unfortunately, some very enterprising Tropilaelaps mite just decided one day, you know what? I want to see the world. And it kind of struck out on its own. It stopped just feeding on the Asian bees and transferred over to Apis mellifera. And because Apis mellifera is managed all over the world, this organism has been able to move from country to country to country. And that's the reason why it's kind of got people up in arms, because we're worried about where it could end up next. So we have seen them start making their way out of Southeast Asia. They're found in other regions of the world now. So South Asia, they are now found in Korea, where they were not found before. They have relocated to areas of Bangladesh and India and seem to be making their way toward the Middle East. And so there are concerns about the ability of this mite to move from country to country to country. And one of the greatest concerns that we've seen there is that these mites seem to be finding ways to just kind of undermine our expectations of them, because we thought that these mites would not be able to deal with winter. That was our saving grace. We said Varroa was able to spread because Varroa are able to deal with cold temperatures, because they can live on the population of adult bees when there is no brood in the colony. Tropilaelaps doesn't have that ability, and so Tropilaelaps will not be able to survive winters. We will be totally fine over here in the US with our frostiness for a substantial portion of the year. We won't have to worry about these parasites. And then they moved into China and Korea, and we started getting really nervous because they weren't supposed to be able to do that. Apparently, they didn't read any of the papers that we wrote about them. They had no idea what the rules were. And I think that now that we have seen that this organism is adjustable enough to be able to thrive in Korea, in China, we really need to be thinking through how we would handle them potentially showing up in our world.
Do you know how they're surviving the winters in the northern latitudes?
We don't know yet. So that's one of the questions that I was over there to better understand. These organisms are absolutely fascinating. So something that I've been trying to understand in my time there, I've had a few questions I'm attempting to answer. How are they surviving the winter? That's a big question because this parasite is pretty wild. One of the things that's rather wild about it, if you go back to, if you were listening to the first appearance that I had on this podcast, all you Beekeeping Today regular listeners, back in 2018,
November, yeah, November.
Goodness, whew, I was so young back then.
Weren't we all?
But you may have heard me talk about how something that is fascinating when you are a researcher is going back to the earliest papers that have been written about an organism to hear how this creature was described and what are some of the things that when we didn't know pretty much anything about it, what were some of the first things that we discovered about these creatures? Well, if you go all the way back to the very first story of Tropilaelaps, you find that in the scientific literature, it was described as a parasite of rats. And this weird spiny rat mite creature was found on rats and just continued to be considered a parasite of rats for almost a decade before the record was corrected. And I think that is really important to us understanding what is going on with this parasite now, because there are a number of parasites that actually use two different hosts as a means of navigating different elements of the creature's life cycle. Because the bees that this parasite is originally from, so Tropilaelaps was originally a parasite of Asian honeybees, specifically giant honeybees, and giant honeybees are a migratory species. And because these parasites don't hang on well in flight, it seems to be the case that as these bees were leaving, these mites were left behind on the comb. Now, it's possible that during that portion of the year, those parasites were then utilizing creatures like rats as what we call a secondary or alternative host. And so if that is the case, then that may be their means of surviving periods where there is no brood in a colony, periods like the winter. If they're capable of actually leaving, attaching to an alternative host, using that organism as a source of warmth and potentially food throughout a period like the winter, that may actually answer our question. So that's a hypothesis that I've been looking to and trying to better understand this creature, and that curiosity about the earliest elements of an organism's scientific history, I think has served me well enough in the study of Varroa, determining that it's actually feeding on fat body, that I'd like to see if it'll actually serve me again with this Tropilaelaps parasite.
Before you left Thailand, you had certain notions, certain ideas you're going to look at. Mm-hmm. Once you got there and got on the ground and started looking at it, what surprised you the most? What thought changed the most for you?
Oh, also a great question. What thought changed the most for me? Well, Jeff, when I first got to Thailand and started studying this parasite, I had some ideas about its, you know what? When I first started studying Tropilaelaps, I was very worried that this organism, the ways that it infiltrates the brood, the ways that it seems to have this damaging impact on bees without actually without needing the elements of viral components to cause deformities, I was really worried that the bees themselves wouldn't have very effective means of controlling this creature innately available to them. But I've found that the Asian honeybees seem to have a number of mechanisms to control this parasite. And Apis mellifera, even as much as it seems to be vulnerable to parasites in general, these organisms are trying. Like, they are working very hard to find these parasites. They seem to be able to detect them below the capping, and so they're running around and uncapping cells. There seems to be something in them that that knows that there is enough of a problem. It seems almost like the evolutionary arms race here is being fought on both sides, and the bees are trying. And so it is my hope that what I'm seeing there is an emerging evolutionary defense against these parasites. But another problem that I don't think people often recognize is that evolution tends to take a long time, a very lengthy period of time. And so that was the first thing. I guess that's more about the bees than it is the parasites. But that was the first thing that really occurred to me here, is that the bees themselves are trying. And if we can find ways to help them along in this process, that may be helpful. Also, for Tropilaelaps themselves, I think what surprised me a fair bit about them was, so I've developed a means of actually peering into the cells while the mites and the bees are developing. I've created a half wax, half glass cell that kind of allows me to be an eavesdropper on this process. And normally everything that goes on there is kind of a bit of a mystery. All we can do is uncap the cells at different periods and say, after 24 hours, this is what it looks like. After 48 hours, this is what it looks like. But we stop all the behaviors that happen there when we uncap the cell. By being able to peer down into it and look at what's going on into the cell itself and actually record those behaviors, you start to see some really fascinating things here. And something that really struck me about Tropilaelaps is that it actually doesn't seem to be that great at its job. The most surprising thing to me about Tropilaelaps is that it doesn't seem to really know what it's doing as well as Varroa does. Varroa's life cycle, several elements of its biochemistry are very, very, very well fine-tuned for them to be with bees. Everything that they do is just connected with some element of the bee's development. Tropilaelaps seems to be very early in its evolutionary development on bees. And as a result of that, it's doing a fair bit of trial and error itself. And a lot of times you will see error. When Tropilaelaps go into these cells, a lot of the time they don't produce offspring. There's just something that doesn't seem to be stimulated in them to allow them to actually produce offspring in this process. And there's a lot of waste going on as they go back and forth. Something that I try to acclimate people to when I'm giving talks about parasites is to let them know that there is a difference between a parasite and a parasitoid. A parasite is an organism that feeds on a creature and utilizes it sometimes even as a home, but it doesn't want to kill that creature. What we often think about as a parasite, we're thinking about parasitoids. We think about the creatures from Alien that are described as parasites, but they're not. That is a parasitoid. Any organism that needs to kill its host in order to develop, that is a parasitoid. And so there are wasps that'll fly around, sting a caterpillar, and the baby wasp will develop inside the caterpillar and then burst out of its back alien style. The relationship that you see there is one that is fine-tuned for specifically that sort of connection, but a parasite is a bit different because it wants to keep its host alive so that it can complete its life cycle, so that its babies can potentially use that host as an option. Varroa is a parasite on its original host. In Apis cerana, the original host for Varroa, these organisms have this system generated between each other. They've brokered a deal that allows for, even though it's a parasite, even though it's a problem in the colony, it's not such a big problem that actually leads to the death of these organisms. And you can usually judge how fine-tuned a parasite-host relationship is by how usual it is that this parasite will cause the death of the host. When a parasite is in its natural setting, the way that it's supposed to be, it usually will not kill its host. Tropilaelaps pretty much always kills its host. And it is very bad at maintaining itself within a colony. It overruns the colony. It destroys it. It behaves more like a parasitoid than it does a parasite. And that is a testament to the fact that it has not really developed the fine-tuned relationship that we usually see after years, millions and millions of years of evolutionary time.
It's that off-season relationship it has with rats.
Yeah, potentially.
Wow.
That's a lot.
Well, so you're going back now. You're going back to Thailand. How soon do you think you can get there?
Oh, my goodness. Every time I think that I might be able to go back, there's more and more information that comes out about COVID-19 that leads me believing that it probably won't be until sometime next year where I'll actually be able to go back. Actually having to leave was financially and scientifically a difficult decision to make. When the State Department released its travel advisory and upgraded things to a level 4 travel advisory, I was thinking to myself, you know what? I'm right in the middle of the last round of experiments. I've spent a substantial amount of money scaling up to this huge, this very large size production that we've got here with all of these colonies. I had to buy each one of the colonies myself. I had to buy all this equipment to use on them and these gas detectors to measure the concentrations of the gases inside of the colonies. I had to, it was a lot. And having to cut things in the middle, I had to, give the bees away. I had to figure out what to do with the equipment. I couldn't just leave it in Thailand. So all of this equipment had to be shipped back. And in order to get it shipped back in time, that cost thousands and thousands and thousands of dollars. It was a lot all at once. And so now, in considering going back, it's not just the quick decision of, well, I'll just hop on a plane and take all those things back. Now I've got to pay to ship all of that heavy equipment back to Thailand. The microscopes, the gas detectors, there's special regulations for if you want to ship chemicals. There's a whole different shipping system for all of that. Getting it there, getting it set up again, and buying all the colonies necessary, it's going to run a substantial amount of money that wasn't in the original budget for this project. And so it looks like I'm going to have to figure out how to fund this project again in order to finish the rest of the process. And so not only do I have to wait until the COVID-19 situation gets worked out on the worldwide scale, which could take a substantial amount of time as we attempt to develop a vaccine, I have to figure out the financials of it as well.
Do you still have your GoFundMe up for this?
I do still have my GoFundMe, and I might need to update it and just start over.
Well,
because I may be relying on that sort of help again. I really appreciate that beekeepers got so invested in this process to the point where they felt like stakeholders in this. They were like, our bees, they're important to all of us, whether people know it or not. Those bees are putting food on your table, on everyone's table. And so the ways that beekeepers really jumped in and decided to fund this project, it honestly changed the direction of my life at that point and the way that I think about science, because I love how invested people become in these processes when they have their own set of input, either financially, but when they can also hear about how a project is being built from the ground up. I talked about this on the Beekeeping Today podcast last time about this project, and then heard more people become interested in this and send me messages about how much they wanted to see this project go forward. And I may have to undertake a new round of that, but also there are wonderful options available through things like the National Science Foundation, National Geographic, and other things that I'll avail myself to so that things won't be entirely connected with the fundraising process, which is stressful and time-consuming.
Well, I think the work that you've been able to accomplish in the last couple of years helps to, that in itself is a self-promotion and helps fund going forward. Yeah. So we only have a few minutes left. What are you working on next? Before you go back to Thailand, what do you see you working on in the next year?
Great question. So I am working on 2 different projects at the moment. I'm working on a means of adapting the discoveries that were made about Varroa, feeding on fat body, to figure out how best we can utilize that against Varroa. And so we've been working on better understanding this for some time now. We'll be putting out some research into publication of some elements of that we've been learning about, because the fat body has a number of proteins that the mites themselves need in order to initiate reproductive processes in their own bodies. And disruption of the mite's ability to process these proteins, or even their ability to utilize these proteins in their own bodies, can be an effective means potentially of keeping the mites from continuing their reproductive processes. And if they can't reproduce, well, then their population can't grow and eventually it dies out. So we're working on not only better understanding how the mites do this, but better understanding how we can impede the mites doing this in the future. That's something that I'm hoping can even potentially be adapted towards usage against the Tropilaelaps mites. And so we're looking at that as well. This summer, I am also continuing to work on the work that I was conducting in Thailand, in the US, on Varroa. So as I optimize elements of the system that will allow me to hit the ground running when I return to Thailand, but will also allow for comparisons with Varroa's life cycle. The ways that we originally described Varroa's life cycle, it required a lot of work in opening the cells and looking inside. And then some researchers with the Swiss Bee Lab came up with this amazing idea to take pictures throughout the life cycle using plastic cells. And I've adapted some elements of that system to make it a bit more cozy for the mites, using wax for most of the cell and just glass for the top of it, allowing me to peer through and see inside. But now I want to do that with Varroa and see what elements of the description of the life cycle of Varroa may need to be updated. What can we learn from seeing the entirety of the process of Varroa's development on developing bees that can then be compared with Tropilaelapses, so that we can compare, contrast, and better understand what we're working with in this parasite. So that's what I'm working on now when I am able to go into the lab. But unfortunately, there's not a lot that I can do from home. So really, right now, I'm working on writing up my research so that that can be disseminated to the public, and counting the days until the stay-at-home order is over and I can continue my research in the lab.
Yeah, we're all working from home. Totally off topic. We were talking about it a little bit before. What's new in the music front? You playing? You doing anything? Recording anything?
Well, so what's new on the music front? You know what, Jeff? This might just be the kick in the pants that I need, because I haven't updated my music channels in ages. But while I was in Thailand, I actually recorded some music in Thai.
Oh, wow.
So I love Thai music. I was working on a song. I recorded it, did a little bit of a video for it, and just never put it online, which is ridiculous.
Yeah.
So I should actually put that out there. But I also sang a song that I used to hear a lot when I was a kid, that I would also like to put online. It's one that I sang at... what meeting was this? Ooh, I don't want to say the wrong meeting, but I was at one of the large conferences to actually sing at the banquet and give some presentations. And I sang at the banquet Colors of the Wind. And I really wanted to put that version of it, my own version of it, online as well. And so
we'd be glad to help you with that.
Well, I mean, you guys have all the recording equipment. Like you,
yes, we do.
Can't see at home, but Jeff has got some serious
Jeff is
there.
Jeff is the man there.
We can.
Yeah, well, Mike, you got a nice mic yourself there.
Yeah, we've got some stuff we can use, Sam. We can.
Jeff, can we jam next time?
Yeah, let's do that. Yeah.
Kim, do you play anything?
No, I listen.
He plays the radio.
Gotcha.
Anything else, Kim? We can let Sammy get back to work.
I'm exhausted just listening. What a story, Sammy. Thank you so much for being with us today and bringing us up to date. Once you get back or the next time you have time, ring us up and we will get you back here to finish the story, we hope.
For sure. I really appreciate that, Kim. I really appreciate getting the opportunity to talk more about the research that I'm conducting.
It's been a fun time. Really appreciate it and look forward to having you back, Sammy.
Great.
Thanks a lot.
All right.
Thanks a lot.
Thank you.
Well, I'm at a loss for words. This was pretty amazing, and Tropilaelaps, that's a scary thing.
I'm exhausted just after having to listen to it. I can't imagine having to deal with it on a daily basis. Although some of what he had to say today, what Sammy had to say today, sounded like there's might be something good on the horizon. So I think with folks like him. Looking at the problem, we might have a chance. And the one thing that we want to remind people of, he's got a GoFundMe thing up there, and you can get to it from our webpage to help him pay for his next trip back. And his next trip back has got to be made because we got to solve this problem before it gets here.
Yeah, we don't want those things here.
You got that right.
We were talking offline with Sammy, and I was telling him that there are YouTube videos of the Tropilaelaps on comb, and they skitter. They run around on the edge of the comb very fast. I mean, he did mention about the Varroa kind of lumbers and bumbles along on the larva, and it's kind of like a big papa bear just kind of lumbering along. Tropilaelaps, they're on crack. They're on speed. They're just all over that comb, 100 miles an hour it seems. And the first thing I thought of was just having all of those on my hands, and that gave me the willies. I'll be straight up about that. I wouldn't want those in my hive.
Yeah.
It scared me. I'm gonna have nightmares.
I hope you don't sleep well, Jeff. Well, oh, this was good. I hope people listened and enjoyed it because it was fascinating.
Yeah, it was. It's always a treat to have Sammy on the show. Well, that about wraps it up for this episode of Beekeeping Today podcast. Before we go, I want to encourage our listeners to rate us 5 stars on Apple Podcasts, wherever you download and stream the show. Your vote helps other beekeepers find us quicker. As always, we thank Bee Culture, the magazine for American beekeeping, for the sponsorship of this episode. We also want to thank our regular episode sponsor, Global Patties. Check them out at www.globalpatties.com. And finally, we want to thank you, the Beekeeping Today podcast listener, for joining us on this show. Feel free to send us questions and comments at questions@beekeepingtodaypodcast.com. We'd love to hear from you. Anything else, Kim?
I think that about wraps it up, Jeff. It was a good day.
Yeah, it was. Well, thanks a lot. Stay safe.